The Baltimore Virus Classification System is a fundamental framework that organizes viruses based on their genetic material and replication strategies. This system revolutionized the field of virology and continues to guide research and medical advances today. We'll provide a comprehensive overview of this important classification system and explore how it has transformed our understanding of viruses. The visual on this slide shows a diagram with various types of viruses arranged into different categories, highlighting the diversity and organization of the Baltimore system. This provides helpful context as we dive into the details of how this classification system works and its real-world applications. Viruses are incredibly small, ranging from 20 to 400 nanometers in diameter. They exist on the boundary between living and non-living things. Viruses are obligate intracellular parasites. They cannot replicate without a host. They lack the cellular structure and metabolic abilities of other organisms. Unlike cellular life, viruses have a wide diversity in their genetic makeup. They can contain DNA or RNA, in single or double-stranded arrangements. Early virus classification systems were based on physical structure, the diseases they caused, and the host organisms they infected. Morphological classification looked at the shape of the virus capsid and whether it had an envelope. Disease-based classification grouped viruses by the symptoms or diseases they were associated with. Host-based classification divided viruses into those that infect humans, plants, or bacteria, bacteriophages. These early classification systems provided a way to organize the diversity of known viruses, but had limitations as new viruses were discovered. Viruses have a wide range of genetic diversity, using different types of DNA and RNA, in various configurations like single or double-stranded, and even segmented genomes. Traditional methods like studying viral morphology alone were limited in explaining these fundamental differences between viruses. Disease-based classification also failed for viruses that can cause multiple syndromes in hosts. As our understanding of molecular biology grew, a genetics-based system became necessary to properly classify and study viruses. This new framework needs to reflect the mechanisms of viral replication to support ongoing research and scientific progress in this field. David Baltimore was a pioneering molecular biologist who made groundbreaking discoveries in the 1960s and 1970s. He was born in 1938 and showed an early interest in molecular biology, earning his PhD from Rockefeller University in 1964. In 1970, he co-discovered the reverse transcriptase enzyme with Howard Temin, which revolutionized the understanding of RNA tumor viruses. For this and other discoveries concerning virus-cell genetic interaction, Baltimore was awarded the Nobel Prize in Physiology or Medicine in 1975. He also proposed a viral classification system in 1971 that remains fundamental to virology 5 decades later. Baltimore's early career, major discoveries, Nobel Prize, and lasting contributions to the field of virology are highlighted in this slide. The Baltimore system is a way to classify viruses based on their genetic material and replication strategy. We'll start by looking at the foundations of this system, including how viruses produce mRNA, the different types of viral genomes, and the strategies they use to replicate inside host cells. First, mRNA production viruses have evolved unique mechanisms to generate messenger RNA for protein synthesis within their host cells. Next, genome-type viruses can have DNA or RNA genomes, and these can be single or double-stranded, and positive or negative sense. Finally, replication strategy viruses use diverse mechanisms to replicate their genomes inside the host, which is a key part of their life cycle. Understanding these foundational concepts will provide important context for the rest of the presentation on the Baltimore classification system. The Baltimore classification is a system for categorizing viruses based on their genetic material and replication mechanisms. It divides viruses into 7 distinct groups, numbered I through 7. The primary classification is based on whether the virus has a DNA or RNA genome. The secondary features look at whether the genome is single or double-stranded, and positive or negative sense. The tertiary considerations examine the virus's replication mechanism and intermediates. Each of the 7 groups has a unique mRNA strategy for expressing its genetic information. Understanding this classification system helps us better comprehend the diversity and biology of different viruses. The genome and replication strategy of a virus are key factors that determine how it interacts with and infects host cells. Understanding the viral genome and replication process can help predict the virus's behavior, including its tropism and pathogenesis. Classifying viruses based on their genome and replication allows researchers to anticipate their mechanisms and group similar viruses for comparative studies. The viral replication strategy and genome features often inform the development of targeted antiviral treatments and vaccine approaches. Knowing the virus's Baltimore classification group and replication process provides important insights to guide research and treatment development. The summary table provides an overview of the 7 Baltimore groups, which categorize viruses based on their genome type. Each group is defined by the structure of the viral genome, whether it's double-stranded DNA, single-stranded DNA, double-stranded RNA, positive sense single-stranded RNA, negative sense single-stranded RNA, single-stranded RNA with a DNA intermediate, or double-stranded DNA with an RNA intermediate. For each group, I'll highlight a few key examples of viruses that belong to that category. Group 1, dsDNA, herpes viruses, adenoviruses, poxviruses. Group 2, ssDNA, parvoviruses, circoviruses. Group 3, dsRNA, reoviruses, rotaviruses. Group 4, plus, ssRNA, coronaviruses, poliovirus, hepatitis C. Group 5, ssRNA, influenza, rabies, Ebola. Group 6, ssRNART, HIV, retroviruses. Group 7, dsDNART, hepatitis B, hepadenoviruses. This comprehensive table provides a helpful framework for understanding the diversity of viral genome structures. The key characteristics of Group 1 double-stranded DNA viruses are a complete double-stranded DNA genome, similar to cellular DNA, that can be linear or circular depending on the virus family. These viruses utilize the host cell's RNA polymerase to directly transcribe the viral DNA into mRNA. Some viruses also encode their own transcription enzymes. Notable members of this group include herpes viruses like HSV and VZV, adenoviruses, and poxviruses. This group includes both enveloped and non-enveloped viruses. Understanding the genome structure and replication mechanisms of these viruses is crucial for developing effective treatments and vaccines. The slide outlines the key steps in the replication cycle of Group 1 viruses, which have a double-stranded DNA genome. It starts with the virus entering the host cell and shedding its protective coating, allowing the DNA genome to be delivered to the appropriate cellular compartment. Next, the host or viral polymerase transcribes the DNA into mRNA, with early genes typically encoding proteins for DNA replication. The viral DNA is then replicated using the host or viral machinery, usually occurring in the nucleus for most Group 1 viruses. Finally, the new viral particles are assembled and exit the cell, either through lysis or budding from the membrane. The animated diagram helps visualize this step-by-step process. Herpes viruses are a group of viruses that cause a variety of infections, including herpes, chickenpox, and mononucleosis. A key feature of herpes viruses is their ability to establish latent infections, where the virus remains dormant in the body and can reactivate later, causing recurrent outbreaks. Adenoviruses are known for causing respiratory and eye infections. Interestingly, they are also used in gene therapy and as vaccine vectors, leveraging their ability to infect cells and deliver genetic material. Poxviruses have large, complex genomes and are responsible for causing smallpox, which has been eradicated, as well as other skin lesions. Group 2 viruses have single-stranded DNA genomes, which are typically circular but can also be linear. Their genomes are relatively small compared to Group 1 viruses. These viruses rely heavily on the host cell's machinery to replicate, so they often infect rapidly dividing cells. Key examples of Group 2 viruses include parvoviruses, like parvovirus B19 in humans, as well as circoviruses that can affect animals and plants. The 3D model on the slide shows the characteristic single-stranded DNA architecture and transparent capsid structure of a parvovirus. Group 2 viruses have a single-stranded DNA genome that needs to be converted to double-stranded form to replicate. They use the host cell's DNA polymerase during the S phase of the cell cycle to carry out this conversion. This requirement often means Group 2 viruses can only infect actively dividing cells. The completed double-stranded DNA then serves as a template for both viral mRNA production and new viral genomes. This replication strategy is illustrated in the images on this slide. The key feature of this group of viruses is their unique double-stranded RNA genome, which is often segmented into multiple pieces. Each genome segment typically encodes a different viral protein, allowing for a diverse set of functions. These viruses carry their own RNA-dependent RNA polymerase enzyme, which is essential for replicating their dsRNA genome, since host cells cannot do this on their own. Important human pathogens in this group include rotaviruses, a major cause of childhood diarrhea worldwide, as well as reoviruses and orboviruses like bluetongue virus. Group 3 viruses have some unique features in how they replicate their genetic material. They create specialized compartments called viral factories in the cytoplasm of infected cells to carry out replication. Their replication process involves conservative transcription, where the original double-stranded RNA genome remains intact while new positive sense RNA strands are synthesized. These viruses require their own viral enzymes, like the RNA-dependent RNA polymerase, since host cells lack the ability to replicate double-stranded RNA. The segmented nature of their genomes allows for reassortment, where different viral strains can mix their genetic material during co-infection of a cell. Positive sense single-stranded RNA viruses have a unique genome structure and replication process. Their RNA genome ACTS directly as messenger RNA when the virus enters a host cell. The host cell's ribosomes immediately translate the viral RNA to produce viral proteins. The viral RNA-dependent RNA polymerase, RDRP, then creates a negative strand RNA intermediate, which is used as a template to replicate the viral genome. This allows the virus to quickly hijack the host cell's machinery to produce more copies of itself. The positive sense RNA virus we're discussing replicates its genome through a series of key steps in the host cell cytoplasm. First, the viral genome is immediately translated by the host cell's ribosomes, producing the viral proteins needed for replication. One of the most important viral proteins is the RNA-dependent RNA polymerase, RDRP, which is responsible for generating the negative strand RNA intermediate. The RDRP uses this negative strand as a template to produce multiple copies of the positive sense viral genome. These new positive strand genomes can then be packaged into viral particles to infect more host cells. The genome of group 5 viruses, like influenza, is made up of negative sense single-stranded RNA. This means the RNA is complementary to the mRNA that the host cell would normally use for translation. The viral genome cannot be directly translated by the host cell. It first requires the activity of the virus's own RNA polymerase enzyme. The RNA polymerase transcribes the negative sense viral genome into positive sense mRNA that can then be translated by the host cell. Some group 5 viruses, like influenza, have segmented genomes, while others, like rabies, have non-segmented genomes. The 3D model on the slide shows the segmented nature of the influenza virus genome and its surface proteins. The slide provides an overview of some key replication complexities found in group 5 viruses, which have negative strand RNA genomes. Viruses in this group carry their own RNA-dependent RNA polymerase, RDRP, enzyme within their viral particles. This allows them to transcribe their negative strand genome into mRNA. Many group 5 viruses also use a mechanism called cap snatching to steal the 5' cap structures from host cell mRNAs. This helps the viral mRNAs initiate translation more efficiently. The viral RDRP can also exhibit polymerase stuttering during replication, adding non-templated nucleotides. This generates protein diversity and helps the virus adapt. Overall, these replication complexities allow group 5 viruses to successfully replicate their genomes and express their genes within the host cell. The slide provides an overview of group 6 viruses, which have a positive sense single-stranded RNA genome and a DNA intermediate in their life cycle. These viruses enter the host cell with their RNA genome, which is then converted to DNA by a viral enzyme called reverse transcriptase. The DNA then integrates into the host cell's genome, becoming a provirus. The host cell's machinery then transcribes the provirus to produce new viral components, allowing the virus to replicate and spread. This unique life cycle involving both RNA and DNA is a key characteristic of this group of viruses. The reverse transcription process is a key step in the life cycle of retroviruses like HIV. This slide outlines the timeline of the different steps in the reverse transcription process. Entry into the host cell happens relatively quickly, within 0.5 hours. Reverse transcription, where the viral RNA is converted to DNA, takes about 4 hours. Integration of the viral DNA into the host cell's genome takes around 6 hours. Transcription of the integrated viral DNA to produce new viral RNA takes 12 hours. Finally, the assembly of new viral particles occurs over 24 hours. Understanding the timeline of these steps is important for developing effective treatments that target different stages of the process. The unique genome of this group of viruses is partially double-stranded and circular, with single-strand gaps. This compact genome structure allows for efficient use of the limited genetic material, with overlapping reading frames. Hepatitis B virus, HBV, is the key example of this viral group, and is a major cause of liver disease and liver cancer globally. Like retroviruses, these viruses use reverse transcriptase, but the direction of information flow is different from retroviruses. The slide outlines the unique replication cycle of Group 7 viruses, which includes DNA repair, transcription, reverse transcription, and virion assembly. DNA repair involves the partial double-stranded DNA being repaired to a complete circular form in the nucleus. Transcription uses the host cell's machinery to transcribe the viral DNA into mRNAs and pregenomic RNA. Reverse transcription then converts the pregenomic RNA back into DNA using the viral reverse transcriptase enzyme. Finally, the nascent DNA and viral proteins are packaged into new virus particles during virion assembly. This cyclical process allows Group 7 viruses to replicate effectively within their host cells. The Baltimore Classification System is based on viral genome type and replication strategy, focusing on viral mRNA synthesis. It has 7 distinct classes, making it a mechanistically oriented system that is practical for research applications. In contrast, the ICTV taxonomy uses a hierarchical classification system that considers evolutionary relationships and morphological features. The ICTV taxonomy includes hundreds of genera and species, and is continuously updated as new viruses are discovered. Both systems provide complementary ways to understand and categorize viruses, with the Baltimore system being more focused on the biology, and the ICTV taxonomy being more comprehensive from an evolutionary perspective. Classifying viruses by their genome type informs the development of diagnostic tests like PCR and sequencing, which are crucial for detecting and tracking viral infections. Understanding the genome type also helps predict how a virus may behave during an infection or epidemic, which is valuable information for public health. Each Baltimore group of viruses has unique replication mechanisms, which provides opportunities to selectively target and inhibit them with antiviral drugs. Knowing the genome type influences the design of vaccines, as different approaches may be needed for RNA versus DNA viruses. The rise of mRNA vaccines has leveraged our growing understanding of viral gene expression to develop new and effective vaccination strategies. Next-generation sequencing allows us to rapidly identify and classify novel viruses, providing immediate insight into their mechanisms. Computational bioinformatics tools can analyze viral sequences to predict their Baltimore classification, enabling a fast response to emerging pathogens. The Baltimore Classification System helps us track viral evolution and host adaptation, revealing patterns in how viruses have evolved different replication strategies over time. Understanding these patterns is crucial for developing effective treatments and interventions against viral diseases. While the current virus classification system is a useful framework, it doesn't always neatly accommodate certain viruses that have ambiguous or complex characteristics. Some viruses don't fit cleanly into the 7 main groups, as their properties may overlap or defy easy categorization. Giant viruses and those with segmented or mixed genome structures can be challenging to classify using the standard system. The current taxonomy also has limitations in fully capturing the evolutionary relationships between different virus types. Integrating the virus classification system with other taxonomic frameworks used in biology can present some challenges that require careful reconciliation. The future of virus classification will involve integrated approaches, combining traditional methods like the Baltimore system with newer techniques like phylogenetics and structural studies. This will allow us to create a more comprehensive classification system that reflects the full range of viral properties. Computational advancements, especially in AI and machine learning, will be key. These technologies can help predict viral behavior from genomic data and automate the classification of newly discovered viral sequences. Metagenomics is also opening up a vast virusphere that we need to accommodate. Classifying the viruses identified through environmental DNA, RNA sampling will be an important challenge. Overall, the goal is to develop a more holistic, data-driven approach to virus classification that keeps pace with our rapidly expanding knowledge. The Baltimore Classification System is a foundational framework in virology that categorizes viruses based on their genome type and replication strategy. This system has stood the test of time and remains a crucial tool for understanding viral behavior and informing research and medical applications. As new viruses are discovered and technologies advance, the Baltimore system continues to adapt and evolve while preserving its core principles. In summary, the Baltimore system is an essential cornerstone of modern virology that will continue to guide our understanding of these ubiquitous and often impactful biological entities.